Awesome. Perfect. Hello, everyone, and welcome to the second webinar of the pre-training for the Scientific Innovation and Translational Research School. I'm Marta Campabadal. I'm the Open Academy Senior Manager, and I'll be your host for today's webinar. I'll do a quick introduction, and then I also want to remind you of a couple of things, and then we'll hand over to today's speaker. First thing is that I hope that you've been making good progress with the e-learning since the last time we saw each other, and also that everyone is preparing for their trip to Barcelona. Just a quick reminder, the program for the face-to-face training has been recently uploaded to the school page. So be sure to check it out to see the start and end times. Before we get started, just a few reminders. If you have questions, feel free to type them in the chat or raise your hand if you would like to ask your questions live or share your experience with natural history studies with the rest of the participants. As you saw, the session is being recorded and will be uploaded to the school webpage later today, along with the slides. Before you leave, please take a minute to fill out the feedback survey that we'll share in the chat later in the session. It really takes just 1 minute and it helps us improve the future webinars. Okay, so going to today's webinar. Today we'll be discussing natural history studies in rare diseases, an essential topic for understanding disease progression and improving research efforts. First of all, a big thank you to everyone who answered the survey on this topic that we circulated a few weeks ago. Your insights and expectations really help us shape this session. We are very excited to have with us Dr. Angela Schulz. She's a senior physician and medical specialist in pediatrics and youth medicine. And she's with us today and will guide us through key aspects of natural history studies in rare diseases and also their challenges. And with that, I'll hand it over to Dr. Schulz to begin today's webinar. Angela, the floor is yours whenever you're ready.
Thank you. Thank you for the nice introduction. And yeah, welcome everybody. It's my pleasure to give today's presentation on natural history studies in rare diseases. And I think we will hear both about challenges, but also I think great opportunities that these natural history studies could provide, especially in the field of rare diseases. These are my disclosures. As you all might know, there are challenges in the clinical trial design and in the design of endpoints in clinical trials, especially in rare diseases. The reason why I already started with talking about clinical trials is that I think this is the ultimate goal when I perform a natural history study in a field of rare diseases. I think that it would be fantastic if data that are collected in a natural history study could ultimately serve as natural history control data in a clinical trial. Because in the rare disease field, many of these diseases do not have any kind of treatment. Many of them are fatal, and therefore it is a good opportunity to maybe have a way to prevent placebo controls in a couple of trials by using natural history data. And therefore, I think one should have that in mind when designing a natural history study. And this is why I'm going to share my experience with setting up natural history studies and how these data could ultimately be used in clinical trials. So I have listed here a couple of challenges that I think are really crucial in the field of rare diseases. So as the name says, rare diseases, we do have a limited number of patients. In many of these diseases, we have a significant phenotype variability. Despite this, we need reliable clinical outcome measures. We also need functionally relevant clinical outcome measures. This is really required by regulatory authorities when such outcome measures are being used in clinical trials. And I think that natural history data can really help finding those outcome measures. And also, I think that there's a way to use natural history control data in clinical trials. This is always a matter of debate. This is why I put the question here, can it be done? But hopefully by the end of today's talk, I will be able to give you a good example that it can be done successfully. If we start with the first challenge, the limited number of patients, I think because of the limited number of patients, really natural history studies in rare diseases require international collaboration, which is why I think the EuroDis effort and this webinar and this school is so important, because it will foster international collaboration, European collaboration among researchers. We have had a very successful international collaboration in a database that is called the DEMCHILD NCL Patient Database. DEMCHILD stands for Dementia in Childhood. And this is a database that was originally funded as part of a European Commission consortium. It was funded under the 7th Framework Program. And we had 5 countries participating in that endeavor. It was Germany, Italy, UK, Finland, and also India. Since then, and this was in 2013 when this project ended, this database has successfully grown into a very significant international collaboration. As you can see here, we have many collaborating centers across the world, and this has really helped us to collect natural history data in these rare diseases across multiple sites. But we have also learned what is important when going on such an endeavor and what are the obstacles. So it's not so much about what kind of diseases these are, it's more about what have we learned from this international collaboration. So the database that we have created is a database that collects natural history data on a certain group of pediatric neurodegenerative diseases. They are called, in brief, NCL diseases. NCL stands for neuronal ceroid lipofuscinosis. But basically, if you look here at the goals of this database, you could replace NCLs by any other rare disease, and I think it would still be true. So the goal of such a natural history database would be to really improve early diagnosis of these diseases. By looking at natural history data, I think this will help us to find early signs of the disease, to improve early diagnosis. And I think we would all agree that early diagnosis is crucial in order to have successful treatment. You want to treat these diseases before symptoms occur or as early as possible in order to hopefully prevent any additional symptoms to occur. We also want to optimize the standard of care for these patients. These are rare diseases, and often these patients do not have access to specialty centers, or specialty centers might still use different standard of cares across the world. So optimizing standard of care for these patients, I think, is really crucial. And by collecting natural history data, and also by collecting whatever is being done under standard of care conditions for these patients, and by evaluating those data, we have a good tool in our hands to optimize standard of care for such patients. Of course, the overall goal is to collect precise natural history data, and here really I would like to stress the point precise, and I will go into more detail in a minute what, in my opinion, precise would mean. And I mentioned that before already, we want to establish evaluation tools and outcome measures for future or current experimental therapies for these diseases. For the DEMTEL database, we were able to obtain ethic approval, and we made sure that whatever we collect as data and however we share these data, that this is always in line with the European Data Protection Guidelines. We were able to have informed consent forms with patients that would allow us to share these data with any kind of third parties, be it scientists or pharmaceutical industry. And in our case, also, we are able to share data outside the European Union, which has always been an obstacle because other data protection guidelines, for example, in the US, might be very different to the ones in Europe. But as we need an international effort in the field of rare diseases and international collaboration, I think it's important to keep in mind that you might also want to share data or collaborate with centers outside the EU, because we really want to support the development of various therapies as much as possible. Also, we think it's important to keep such data collection as independent as possible. So to not have such a database be owned by one pharmaceutical company, but also to have the data independent and be able to share these data with whoever might be a candidate who could make use of them. The collection and the sharing of the patient samples with third parties is equally possible. So we have implemented what we call a virtual biobank. We felt it has been difficult to kind of have a central biobank where all the samples are being stored, because some countries might have different rules for storing these samples than others, but having a virtual biobank might be useful. And if you have a natural history study, you could at least collect data on where samples are present and where are they scored. The structure that we have been using so far is a REDCap system, which we feel works quite well because most sites, most university centers have this system and are quite well versed in using such a system. So it would be good in general if you have a natural history data set or natural history registry to not just use like an Excel sheet, but to have something that's protected, that can be used at multiple sites, and that is always in line with data protection guidelines. So this is why we have a database user agreement among all the sites. We have sites that might add specific items that they collect only at their site because they might have access to this kind of data, and that might not be collected at other sites, but that's possible. And really most importantly is the focus on data safety. So we have implemented an audit trail. So whoever changes data in that database will be recorded, and we could always go back and see when has what been changed. The data storage is on 2 different servers with an emergency power supply, and there is a backup of the entire data set every 24 hours. This data safety really has been crucial in allowing these data to be used as natural history control data in clinical trials. Therefore, I think it's good to have that in mind when you start such a natural history data collection. Our ongoing work at the moment is that we would like to enable parents, because we are a pediatric patient cohort, so parents are important, to also be able to directly feed data into the database. I will come back to that later, what kind of data parents would be able to feed into the database. And we also want to expand this database to other pediatric neurodegenerative diseases, to not have it just disease specific. I talked about the database user agreement, and again, here it's important that the patient rights are well protected, and I think this is important for any kind of natural history study. You need to comply with your local ethic regulations. You need proper consent forms. You have to ensure the data safety. So usually if you have such a database, it should be password protected. You should not use the real patient names, but have patient codes or IDs. You have to make sure that the data quality is really, really high, but also that the data entering center feels and is responsible for the data quality. Ideally, it would be good to have somebody independent who is able to monitor the data from time to time. Monitoring the data would mean that whatever is being entered into your natural history database or registry is being source verified. So you need clinical sources, be it questionnaires, discharge letters, results from medical charts that are available and that are proof that the data that you're entering into your database or that you're collecting into your natural history study are true and actually there. Because regulatory agencies, if you use those data later on in clinical trials, might come to you and might want to see these sources. So it's very, very important that these sources are maintained in a high quality state and that they are monitored. And there's always a question about data ownership. In our experience, it was always best if the data entering site remains the owner of these data and can decide whether or not to share the data with other sites. We also have good experience in differentiating between two types of data that we are collecting. Our database and our standard of care with these patients allows us to see these patients in a prospective longitudinal way. This might not always be the case in a natural history study. There might be natural history studies where you have to collect your data retrospectively. It might be that you do cross-sectional data collection where you see or where you collect data only once in each patient. But if you have a longitudinal natural history study, I think it's important to look what are what we call static data and what are dynamic data. Static data are data that are not going to change in the future anymore. They are really bound to a certain age or examination time of the patient, and you could collect them also retrospectively by using patient charts, by using, in our case, because these are pediatric patients, parent interviews, but you could also, in adult patient, use patient interviews. This is here example on what the database looks like. For example, static data would be data on the gender of the patient. These would be data on the diagnostic summary. What kind of disease has the patient? What is the genetic diagnosis? What is the medical history? What happened prior to diagnosis of this disease? Here, for example, we see the respective mutation in a patient, but also here you see an example of what we call our virtual biorepository. We ask whether there are any patient materials available. For example, here you can see that fibroblasts are being ticked, and we can also see at what center, the center in Hamburg, these specimens are available. So if somebody looks at the database, they could then get in touch with the respective center and ask for these samples. And I think this has also been initiated because we're dealing here with the pediatric population. So repetitive invasive sample collection, especially in pediatric patients, should really, really be prevented. This is why we are collecting this information in addition to other natural history data. As we are performing longitudinal data analysis, we have a second set of data that we call the dynamic data. Dynamic data, in our case, are collected prospectively. So they are always related to the patient's age and the date of examination. You see here a long list of different types of data that can be collected in our database. I'm not going to go into detail. This is really disease specific. But the important part is that you choose a date on which day this assessment has been performed, and the respective assessment then are being collected in an ongoing fashion. You can see that here as well. So if we look at our challenges for natural history studies in rare diseases, the limited number of patients as a challenge might be well overcome by international or European collaboration. If we go to the next challenge, the phenotype variability. Some diseases that we are dealing with have a very significant phenotype variability. And please allow me to give you a little bit. So if we look at this phenotype variability and we look at different assessments that could be performed in order to illustrate the natural history of this disease, we see some of these assessments, for example, those that look at gross motor or fine motor function or language function, cannot be performed in the infantile phenotype, but can well be performed in the juvenile patients. The same for cognition. The infantile patients are too young, they're too rapidly progressed. We cannot really perform cognitive assessments. If we look at the age at which key symptoms of the disease start, and we compare this between the infantile and the juvenile phenotype, you see also that there's such a significant difference. If we start at the age of vision loss in the infantile phenotype is at around 19 months of age, in the juvenile phenotype at the age of 11 years. Also, end of life, patients with infantile phenotype might get around 9 years of age. However, juvenile phenotype patients can get as old as 34 years. So we have a very, very different disease duration, spreading a few years up to almost 20 years. All this would really, really influence your natural history study. And again, if we look at potential tools for outcome measures, you can see here, I will not go into detail of these tools because these are very specific pediatric tools that we use in pediatric neurodegenerative diseases. But if you just compare infantile and juvenile, you see that for many tools, they cannot be used in the infantile phenotype, whereas they can be used in the juvenile phenotype and vice versa. So you cannot do one natural history study using the same tools in both phenotypes. You might have to decide which phenotype you want to focus on. And you see that here in a couple of these tools. So can we overcome these obstacles? And we think they can be overcome, but we really need to look at the challenges both phenotypes cause when looking at a clinical trial. The juvenile phenotype allows us to really do developmental testings because neurodevelopmental milestones are reached in all patients. So we can use clinical scores and test batteries like neurocognitive testing in these patients. And therefore, in this phenotype, already quite large and valid natural history data are existing. However, if you look at clinical trials, therapeutic trials, this disease has the challenge that it's very slowly progressive. So we would need a long trial duration in order to be able to see efficacy of an experimental treatment. And that's something that causes many of the pharmaceutical companies to shy away from doing clinical trials in such a phenotype because it's very costly. If we look at the infantile phenotype, there is the advantage, so to say, even though it's a bad advantage, that it's fast progressive, and therefore we could see a potential efficacy of an experimental treatment quite rapidly. However, in these patients, most developmental milestones are never reached. So they never learn how to stand without support, they never learn how to walk, the developmental level is very low, and therefore clinical scoring systems that we use in other pediatric patients cannot be used in these patients. Which is why we decided that we need to develop a very, not only disease-specific, but disease phenotype-specific rating scales for this respective disease in order to quantitatively describe the disease severity and progression in this infantile phenotype. And I would like to give you that example because it might be the same for other rare diseases. You might need a disease-specific scoring system in order to have good natural history data. And you might not even to be disease-specific, but also phenotype-specific. The requirements that we wanted to cover are that such a scoring system should be easy and quick to use. You always have to think about the reality in everyday clinic. If you have an assessment that takes an hour or two or even more for the investigator to use, it will be hard to have a large number of natural history data because it takes so much time, and then it will be hard to have enough people to really be motivated to use this. Also, it's always good to have a tool that you can use both retrospectively, but also prospectively when you continue to see the patient. It's important that you have things that are functionally relevant outcome parameters. This is something that is always being required by regulatory authorities. They do not only want to see whether, for example, a neurologic reflex is still there or not there. They want to see whether the outcome measures that you take are really important for the patient's daily life. And it's important with all these assessments that you do that they have excellent inter-rater reliability. So the way we started to do this for this infantile phenotype was if that first of all we would ask the question whether there's a regression of function. If that's not the case, then we would have to look at the question whether developmental level of this child is normal. If that's the case, then a child would get a full score, for example, of 3 points. If the developmental level is not normal, then there's loss of 1 scoring unit in that respective function. If there's already a regression of function present, then the question is whether independent active function is still present. If yes, it's a score of 1. If independent active function is not there anymore, then it's a 0. So we kind of differentiate in the different functional categories whether a patient has normal function, whether there's a delay in the functional development, whether there's a regression of function, and whether there's a loss of function. It's a very, very simple way to score a patient. It's not complicated, and it shouldn't be complicated so that many people can use this type of assessment. And we looked at different functional domains: gross motor function, fine motor function, and expressive language. And you see here in each of these categories, if you look at the definitions, we always look whether a function is normal, so age appropriate, whether there's a delay, then there's a loss of a scoring point, whether there's a regression in this function, there's another loss of a scoring point, or whether there's a complete loss of this function, so then the score would be 0. So if you put these 3 domains together, you can have a maximum of 9 scoring points if everything is fine. Very easy. In this specific disease, we also had additional meaningful categories that were clinically meaningful, where we also just differentiated between a score of 1 for age appropriate and 0 for pathologic. Very, very easy and straightforward. So if we now look at our patients, we had a total of 14 patients with this phenotype and CLN1 disease that we were able to score longitudinally over time. You see here in this graph the age of the patients and the total score of the main functional domain. So the total score was 9, 3 times 3. And we can see here that these 14 patients show a very high homogeneity of the loss of function. So the gray area, which is the 95% confidence interval, is very narrow. If we look at the individual scores here, the full score that also has the add-on categories, again, you see this steep decline, this steep and rapid loss of scoring units, but you also see that these 14 patients behave very, very similarly. And this shows us that the scoring system also works quite well. So we have a good score in our hand that we can use in order to collect natural history data that also shows very well how this phenotype presents. So different phenotypes, in our opinion, require a very phenotype-specific outcome measure. We cannot use the same outcome measures across different phenotypes. So what kind of outcome measures can we use? They should be reliable. I already mentioned that they should be inter-rater reliable. But there are also other outcome measures that we can use that should make sure that they are not being influenced by the respective rater. Again, coming from the pediatric neurodegenerative disease field, we use imaging and computer-based clinical evaluation tools. For example, we have a way to analyze gait of these patients with an electronic gait analysis system. So patients would have to walk across a mat that you can see here on the left side. And this is an electronic mat that looks at different gait parameters of the patient. For example, on the graph here on the right side, you see the correlation between gait velocity and gait cadence in our patients. And this is shown in different colors here. These are patients who have different impairments of their motor function. And you see that patients with green have a good correlation between velocity and cadence, patients in yellow and patients in red. So this is a good way, a very objective way to analyze gait in our patients. Another way would be, these are neurodegenerative diseases, to look at their magnetic resonance images. So at the images of the brain of these patients. Their brain volume shrinks over time when the disease progresses, and we can analyze the volume of the brain, and we can find out that this is also a good parameter to illustrate disease progression. So computer-based and imaging tools can be an important part of your natural history studies because they are independent of the different assessors, other than, for example, a scoring system that might always be somehow a little bit influenced by whoever is using this. So if you use scoring systems and other assessments, clinical assessments, you have to make sure that you train the investigator properly, that everybody does the same thing. Another imaging evaluation tool are, for example, also ophthalmic imaging tools. This is the ocular coherence tomography. With this tool, you can measure thickness of different cell layers of a patient's retina. We are using this because in our patients, vision loss is one of the main symptoms, and this is vision loss due to retinal degeneration. You can see here on the left side from A to F how the retinal thickness becomes thinner and thinner over time. And we can see here in the graph as well, where you see the retinal thickness according to age, and you see that the individual patients lose retinal thickness over time. And this is one of the outcome measures that we are using. So these are outcome measures that are not influenced by the assessors, but also we have to keep in mind that patients are taking different current medications, that patients might have different palliative care measures. So your outcome measures should not be influenced by these things either. I mentioned before that outcome measures should also be of high functional relevance. This is something that regulatory authorities keep asking us. And in order to find such outcome measures, we think it is important to involve also the patients, the parents, the families. And the way we would do that is something that we call PPPI. PPPI stands for Patient-Parent Public Involvement. And it's really well described by the term nothing about them without them. This becomes more and more important. Research should be involved with or by the patients, and in pediatric patients, the parents, but also members of the public, rather than just be about the patient. And I think it's especially important in the rare disease field. We should really include the recipients of the healthcare, of the treatment, of the medical care and the social care. So PPPI is a very important cross-cutting issue, and I think it's important to involve the patients and ask them what is important. What is important in your disease? What kind of data should we collect? And this can be really, really helpful. So when we ask our patients and parents with regard to our database, what is important? What do you want us to focus on? Their feedback was that they want the data that we collect to be used as controls in clinical trials because they do not want the children to be treated with placebo controls. They also want us to really, really collaborate with regulatory agencies early on so that any data that has been collected can be used in the future in natural history trials and also in clinical trials that develop experimental therapies. They want us to collaborate with pharmaceutical companies to really make the most of these data, to not have to collect the same data over and over again, and also to use the time now when maybe a treatment is not available yet. You can also use the time to collect natural data, history data now, to gain time and then be ready and have these control data ready for use in experimental clinical trials. And parents really want to have the possibility to directly feed data into the database themselves. In that case, it is important that we really define what kind of data are being entered into the database by either patients or parents in our case. Do we mean the same thing? Are the questions that you ask for your natural history data really well defined? Do you mean the same? Does the clinician mean the same when answering such a question compared to parents or patients? And also it's important to ask the patients, do we miss anything important? Do we miss an important symptom of the disease? Is there anything that bothers you in your daily life that we are not asking by our questionnaires or that is not being assessed by the different clinical scores? So it's important to really have a very open and productive discussion with parents and patients. And again, because here we are talking about a pediatric population, but I think it's the same also in adults. Nobody wants to have repetitive invasive sample collections. So whenever you have samples, it would be great to collect the information. Where are the samples and how can they be made available? And again, I said it before, what are patient relevant data? How can clinical scoring systems be improved to really ask what is important about the disease? What is really bothering the patient? So in order to develop functional relevant clinical outcome measures for your natural history studies, it's important to involve patients in the pediatric population, the parents, and also the public to really find the best outcome measures. This leads us to the final question, looking at all these challenges. You could say, well, is it even possible to use natural history control data in clinical trials? Can this really be done? And I would like to give you an example that we managed quite well in one disease to really use our natural history control data for a clinical trial. So if we look at our lessons learned, international collaboration. I'm now giving you an example for a disease that is called CLN2 disease. This is a pediatric neurodegenerative disease where patients have symptoms quite early in their life. This starts already at the age of 3 to 4 years, and it's a very rapidly progressive disease. Patients lose their function within a few years, and their life expectancy is clearly reduced, and they would, untreated, only live for about 10 to 12 years. Scales pretty much have the same definition of motor and language function in our entire cohort shows the following. So first, we looked at the data in our DEMCHILD database, which were longitudinal data, and we looked at the sum of the motor and language score across time. So each category had a maximum score of 3, so the sum would add up to a total score of 6. And this is shown here in this graph according to patient age. And quite similar to the other scoring system that I showed you for the infantile CLN1 disease, also here we see that the patients lose their motor and language function very rapidly, but also in a very homogeneous fashion. Again, the 95% confidence interval is very, very narrow. If we now put additional data on top of this graph, and these are data from the Weill Cornell Medical College in the US, which were cross-sectional, so this is why we have individual data points here, we see that they really fall quite well within the 95% confidence interval of the longitudinal data. So longitudinal and cross-sectional data match very well in our case. Ideally, because this is a regressive disease, and we want to really assess the rate of disease progression in our patients, data should allow to do so. And if we calculate the rate of loss of scoring units in our patients, we come up with a rate of decline of about 2 scoring units per year in our total of 41 patients. So we can calculate the speed of disease progression in these patients. So we were able to really show these data to regulatory authorities, both the European Medicine Agency, EMA, but also the US-based Federal Drug Agency. These agencies then came to the respective data collecting sites that were part of this natural history study, and they audited us. They really wanted to see whether this natural history studies were of such high quality that the data could be used as control data in a natural history study, but also in a clinical trial to be used as control data. When they performed these audits, it was important to be able to show that any data that you would have in your data collection could be well cross-referenced with data from medical charts. So if you, for example, collect data using questionnaires, that you give the parents or your patients, it's important that you also have medical charts that corroborate and support your data collection. So it's usually not sufficient to just have a questionnaire being answered by a patient. It's important to have medical charts where examination results really show similar description of symptoms. Also, these regulatory agencies really focused on the data safety of these natural history studies. So they really wanted to see an audit trail. Data safety does not only mean that others cannot access the data and that patient privacy is really maintained, but also data safety means that not somebody can later on go into your data set and change the data on his own behalf. So the audit trail is super important. You have to show if data have been changed later on, who has changed them, why have they been changed, and this has to be tracked. Also, it's important that the data storage is very safe, so you need your own power supply, and that the data set is being backed regularly, ideally every 24 hours. So we use those data in as natural history control data in a clinical trial. And I said before that this new treatment that was checked in this clinical trial was an invasive treatment, which is why we really wanted to prevent placebo controls. The treatment entailed that the medication, which was a recombinant enzyme that patients could not produce due to that genetic defect, that this recombinant enzyme has been applied into the ventricles inside the brain. This is a neurodegenerative disease, so the patients need this enzyme inside their brain, close to the neurons, to be uptaken by the neurons. So this enzyme is called TPP1, and we're able to produce the recombinant form of the human TPP1 enzyme, and this needs to be applied into the brain. Because it was quite a large protein with a molecular size of 55 kilodalton, the enzyme was not able to cross the blood-brain barrier, which is why it needed to be applied via what we call a RICAM device. A RICAM device is a reservoir that's placed inside the skull of the patient, underneath the skin of the head, and this is then connected with a catheter into the ventricles, so the inner rooms of the brain. And you can use this device in order to infuse the enzyme from the outside into the inner side of the brain. And this is repeatedly done every 14 days with an infusion that lasts for about 4 hours. You can easily imagine that this is quite an invasive treatment for these children, and using a placebo-controlled infusion in these patients would be unethical. This is why it was so important to have good natural history data in such a clinical trial. I already introduced you to this motor and language score that we used, the total score of 6. And this was chosen as a primary efficacy endpoint in this clinical trial because we had good natural history data showing how rapidly untreated patients would lose this score. And these data were ready to be used already at the beginning of the trial. And you see here on the right side, this primary efficacy outcome in this phase 1/2 clinical trial. So again, because this was a rare disease in a pediatric population, we had to start the phase 1/2 trial in the patients already. So we did not have any adult volunteers who were treated with this enzyme. This wouldn't have made sense because a normal volunteer would be able to produce this enzyme and would not show any side effects. If you look at the graph here, we see the loss of this motor and language score over time. And in red, you see untreated patients. These are our natural history data, where patients rapidly lose motor and language function. And in blue, you see patients that are treated with this enzyme every 2 weeks. And you see that patients who are treated with the enzyme are much more stable over time. They do not lose their motor and language function as rapidly. And because of these data, we had a very rapidly approved treatment for this otherwise deadly and devastating disease. We treated the first patients early in 2014, And we had approved treatment both by European Medicine Agency and also later by the FDA in 2017. So a very rapid trial. And also we only had 24 patients treated in this trial. So we had only a small cohort of patients and that were treated. And again, this was possible because of the natural history data. So I think this trial shows that independent natural history control data can be used in order to advance therapy development in rare diseases. And if they can also, if collected early on, they can speed up the therapy development. So if we look at this specific CLN2 disease, you see here our first target was the preclinical testing of an animal model, which in our case was a naturally occurring dog that has the disease, which happened in 2010 until 2012. The collection of the natural history data had started way earlier. We started already in the year of 2000 to really longitudinally collect these data. At that time, we did not have any idea when and if at all one day we would have an experimental treatment for this disease. But nevertheless, we felt we need to start to collect these data, and it was proven that this was a good decision to start early on. So in 2013, the first clinical trials started, and in 2017, we had the first drug approved. And now, at the moment, we are able to even treat patients early, pre-symptomatically, and we are about to start a newborn screening program for this disease because we really want to prevent patients to become symptomatic at all. We want to diagnose them right after birth and start treatment right away, which is the final goal for such a devastating disease. And I hope that with this example, I could show you that natural history studies are super important in rare diseases and that they have a very, very important role in the development of experimental therapies for these diseases, but also that they have some challenges. You have to make sure that you have high quality data. You have to make sure that you choose the right outcome measures when you collect these natural history data. And you have to make sure that data are being monitored, that they can be audited by regulatory authorities, and that you really do it in a way that international collaboration is possible. And with this, I thank you for your attention, and I'm happy to take any questions.
Thank you, Angela, for sharing this successful case study with us. I'm sure everybody has learned a lot from it. I have one question from Pablo in the chat. I also have a couple of questions that another participant sent me privately, but because he had to leave, I'll ask them myself. Okay, Pablo's question is, as a small organization, we may not have the resources and partnerships to produce a natural history study as complete and multi-layer as this one. Do you think it would be valuable to generate some knowledge through a retrospective natural history study for ultra-rare diseases that has not researched while we gather funds for a prospective natural history studies, or is it better to wait and do it both together?
No, I think retrospective data collection is of high importance because we can learn already a lot from it. I think it requires some time and diligence to really go through these patient charts retrospectively, but we have done both. We have started with retrospective data analysis, and then we continued to collect data also prospectively. So I think there is a high importance of retrospective data collection, and especially in rare diseases, I think we need to also use retrospective data collection because otherwise time will take too long and funds are not there to always just do prospective data collections. Marta, I
You're
cannot.
mute, Marta. Yes.
Sorry, I'm just saying that I pasted a couple of questions that I received in the chat. But if anyone else has questions for Angela, or if anyone else here has experience participating or in a natural history study and is open to share their experience, just raise your hand. All right, first question. How can patient advocacy groups contribute to the design and implementation of natural history studies to ensure they address real patients' needs?
Well, I think patient advocacy groups and patient family associations have a very important role in natural history studies because they can help identify patients that could participate in such a natural history study. They could identify centers and sites and clinicians who could participate, but also they can support clinicians to really use the right outcome measures because they have experience with the disease and they can guide us and tell us, okay, this is really what you should follow throughout the course of the disease because that's a functional relevant outcome measure.
Thank you. And the second question is, what are the biggest challenges in collecting long-term data for rare disease natural history studies, and how can patients and families help overcome them?
I think one of the biggest challenges is really that for something like that, patients would have to come to the respective center who's collecting the data again and again, which requires many cases traveling, which requires time and dedication. And often, at least in our case, patients have dedicated themselves to help us collect these data. And by the time, finally, years later, an experimental treatment was available, these patients might have already passed away or might have been too progressed already in order to fulfill inclusion criteria. So really, this is an endeavor where everybody can support development of treatments, but unfortunately, often patients participating in natural history studies might not in the end later on be candidates that can participate in the experimental treatment as well. So I think one should always think about this and maybe use the natural history data not only as control data in an experimental treatment trial, but also in order to help the patients who have the disease right now and who might only have access to palliative treatment to guide them with regard to other treatments, to other symptomatic treatments. These data can be used also in order to assess what palliative care measures work best, what supportive therapies work best, so that they gain information from this that helps them in their day-to-day fight with the disease. And this requires a lot of time by the clinicians to not only study the disease, but also care for these patients. I think the term care and study is really important so that we do not only kind of use the patients for collecting data, but that we also use the expertise that we gain from these data and give it back to the families.
Good. I don't see any more questions, just a comment from Christian saying that it's very interesting that the history, oh, other one, okay. It's very interesting that the history data can be used to skip the placebo arm of the trial in the case that you shared. And then Crystal asking, no, sharing her experience. I participated in a history study on limb girdle muscular dystrophy. This study is conducted by the Jane Foundation, and they came to Chile to train doctors. I have to go once a year to the clinic where they perform blood tests, spirometry, and various exercises to measure my muscle strength. They also conduct a walking test, and it has been going on for 3 years now. So that's very interesting and
Yeah,
shares.
I think it's, yeah, and it's really, really great that patients dedicate so much time and effort and travel time for these studies. And this is fantastic. And without this patient support, this would not be possible. I think this is very, very important. And therefore, I think it's super important that patients also guide us in what data should be collect and what should we do, and that we inform patients, for example, why do we take these blood tests? What do we do with these samples? What's going on? So we should really have them participate in this endeavor and learn from it as well, and
Yeah.
get results that they can take home and discuss with their own doctors and use them as they might influence their current treatments.
I was wondering maybe if Christine could maybe share how did she find out about this study? If she was invited to participate, if there was I don't know, an application process, if it was referred by her medical practitioner. Out of curiosity, if you want to comment on the chat, that'd be nice. Um,
Hi,
yeah,
can you hear me?
I can hear you. I don't see you, but I can hear you. I don't know where you are, but
I was referred by my neurologist.
mhm,
He is part of the Jane Foundation.
mhm.
So I subscribe to the patient registry in the Jane Foundation, and the doctor uh, is trying to show to the foundation that Chile has a lot of patients with limb-girdle muscular dystrophy. So he was able to bring this natural study to our country, and that's why I participate in the study.
Thank you. Yeah, thank you. Very interesting. Carmen is asking about the outcome measures. Do patients take part in the question process elaboration?
Yes, I think that's really important. Patients took part in establishing these outcome measures and in also how we would phrase the different categories for scoring. I think that's really important, and we learned so much from our patients when doing that. So I think really this has to be an endeavor where all parts are seen and where not just the clinicians come up with an idea how to do something, but they should really listen to the patient voice and ask them whether or not they feel that this makes sense. Because they're really the experts of the disease.
Yeah. Sandra is asking, what do you think about the specific scales to help guide the outcome measure?
Well, as I said, I think this is one of the maybe most difficult, but also most important decisions that you have to make in your natural history study, what scales you want to use. And it would be great to really discuss this not only within your center, but with your patients and hopefully also across multiple centers, because I think it's important to have a good specific scale that really captures symptoms of this specific disease, but also use a scale that is not so complicated to be used, that doesn't require days and weeks of training of the different investigators, but that can easily be used across different sites so that many people participate in your natural history study.
Good. Are there any more questions for Angela? Now is your chance. No? All right. Okay. I think that we can finish here. We can finish a bit earlier. That's good for everyone. You can go and have lunch. Thank you, everybody, for joining us today. Thank you, Angela, for your presentation and for sharing your experience with us. It was very, very valuable to everyone. Just a couple of things before we finish. Take a minute, please, to fill out the feedback survey. The link is in the chat. It's also on the school page. And I'll see you for webinar 3 on the 29th of April on drug repurposing. So thank you, everyone, and see you soon. Bye-bye.
Thank you. Bye.
Thank you.
Thank you, everyone. Bye.
Thank you.
Thank you.